Okay, now I'm going to kick off this with a sort of a brief introduction, which talks about the philosophy behind our extraction methods and some of the reagents that our company has been developing over the last 20 years, and give you a little bit of history behind why we're doing this and why New Zealand was particularly well placed to do this kind of work. Joe will then follow on from me and give you the meat of the seminar, and she'll talk about the sequencing methods that we've been using. So New Zealand is a great place to do work on extremophiles. We're on the Pacific Ring of Fire. We've got alpine areas, we've got volcanic areas, and in addition, we're in quite close and easy proximity to Antarctica. We have regular trips going down there to Scott Base and McMurdo Base, and that gave us the opportunity to study and to collect extremophilic microorganisms. So you can see a few shots from Antarctica. The top left is Shackleton's Hut, the top right are the Antarctic dry valleys. So these are extremophiles, not just in temperature, but they're in extreme dryness. They're xerophiles as well, so some really very unusual organisms. We have hydrothermal springs and vents in New Zealand. The central one there is in the central North Island. And we also have volcanoes. So you can actually see the skiers on the volcano on the bottom right-hand picture, and you can see the eruption of the volcano that they were on, which brought the ski season to a really rather rapid closure in 1998. The middle picture at the bottom, of course, is the necessary hero shot from whenever you go down to Antarctica that you send to your mother. So Antarctica is a very fascinating place, at least biologically. We were working in the dry valleys of Antarctica, so you have these valleys that are as big as the Grand Canyon with glaciers tipping over the edges. But the area to which New Zealand has greatest access, which is the Ross Sea region, not only has the cold environments, it has some of the most isolated volcanoes in the world. So you had the opportunity to find thermophiles, which seems a bit ironic for Antarctica, but thermophiles that have been isolated for millions of years from the rest of the world's ecosystems. So this gave us an opportunity to look for new organisms, and this started out as an ecological exercise when I was in academia at the University of Auckland. Other work that we were involved with was in Scott and Shackleton's historic huts that are in the area around the Ross Sea region. These are still standing, there's still food. You can see the Huntley and Palmer's biscuits there from England, which a name which I think if there's any English people on board, they'll recognize. So there was an opportunity to get organisms, and in this case we were looking at fungal organisms that were growing on these artifacts, and we were sponsored by the by the Historic Places Trust. New Zealand looks after these huts, and there was some level of decomposition that was going on with the fungi there, so they wanted to know what it was and how it could be at least slowed down. Those organisms became useful for some of our biotechnological processes. So you can see this is the meat locker. So if you want proteinases, you'll find them in the meat locker. There's a chicken there and there's a pig there. There's also a dead seal that they've been using the blubber for to do the cooking. So we had lipases, proteinases, cellulases, hemicellulases, all that were coming from organisms that were growing at extremely low and unusual conditions. So what we gained over the years was a very substantial and varied culture collection. It's largely fungi, but we have archaea and bacteria. The archaea and bacteria are largely thermophilic. The fungi are almost entirely psychrophilic, so cold-loving organisms. And we became involved in a number of biotechnological programs. In the initial stages, we were funded by Shell Oil. In the 1980s, they were looking for ways of making biofuels from cellulosic waste, which, if anybody's worked in that area, they will know that it turned out to be not economically or really technically feasible to do that, at least not yet. But it allowed us and it funded us to gain this very large culture collection of unusual organisms with unusual activities. And after that work finished, we started to look at other areas that we could work in. We'd done a lot of work in industrial applications, and we decided to work down at the other end, which was the fine chemical, the low volume enzymes, and they have the advantage that these are enzymes that are used in small quantities and can be used and can be manufactured without needing expensive plants. And the area we looked in was in molecular genetics, and that was primarily because that was the area of our own expertise. I think the picture on the left will trigger a few memories from anybody over the age of 45. When I first started doing my PhD, there was no cookbook for molecular biology. What we had was you had to forage through papers, there was buried information, missing information, there was obfuscation, there were protocols that didn't work. And then suddenly, like this is Prometheus handing down Maniatis and Sambrook to an exhausted molecular biologist. We had a cookbook. This was the Jamie Oliver of molecular biology, and you followed it like it was some sort of scriptural text. If it said you incubated for an hour, you incubated for an hour, even though the method probably developed from somebody that needed a lunch break. You followed it down to the T, and the reason you did that was because most of the time you're working with tubes that appear empty. You can't see your sample, you're working on them for days, and sometimes after 3 days you found that they genuinely were empty all the way along, and you had no way of knowing until you did the final diagnostic. So we became tied into this dogmatic way of viewing things. And that hasn't changed, even though people don't use that book anymore, and not many people, I assume, use that book anymore. Most of those methods that are in that book, in that manual, are still ingrained in the methodologies that we're using. And often, being tied to this dogma led to a set of procedures that were overly complex, unnecessarily so. This was one of my childhood favorites, Professor Brainstorm. So in one area that we're interested in and we're going to be talking about in this seminar is how to get DNA and RNA out of biological material. And the enzyme that is generally used in all cases, and it was used back in the time of the Maniatis manual, is proteinase K. Now, there's nothing wrong with proteinase K. It's a very powerful and effective enzyme. It works in really quite harsh conditions. You can crank up your solution with large amounts of detergents like SDS, and it still works. But there is one problem with it, and that is that it works at relatively low temperatures. So it works best at around 52 degrees C. Some people use it at 65 degrees C, but it really has no activity at that temperature. It's fried egg at 65. So because it doesn't work at that temperature, you don't get the advantage of the heat denaturation of the substrate, which is the protein. And that makes it have a relatively low specific activity. So to improve that specific activity, we add detergent. And the detergent in most cases is SDS, though there are others that are used as well. And as soon as you add SDS, your problems begin. So let's have a look through the traditional way of doing it. I could have picked just as easily a column-based method or a magnetic bead-based method, but essentially we're doing the same set of processing in slightly different manners. So we're getting DNA out of some biological material, bacteria, soil, or a buccal swab, or something like that. And we add our proteinase K with our surfactant, SDS. And then the first step we would use in the past would be to do a phenol extraction. Now, a lot of people believe that that was to remove the proteinase K, but you don't need to remove proteinase K. It can be easily heat killed. At 65, it just dies. But the SDS has to be taken out of the mixture because that's going to cause you problems downstream. You then do a phenol chloroform extraction, and then you do a chloroform extraction to remove the phenol. And then you do an ethanol precipitation to remove the chloroform, and then you wash the ethanol precipitate in 80% ethanol to remove the sodium acetate, and then you dry it down to remove the ethanol. And if you look at all of those steps, every single one of them was to remove something that was in there that you put in the previous step. And it all led back to this one thing, SDS. You had to remove the SDS. So we decided that the problem with these methods was the proteinase case. So we were going to find something a little better and more appropriate for the task. And the one we chose came from Mount Erebus on Antarctica. Mount Erebus is one of the few volcanoes that has an open lava lake in the crater. It's on Ross Island. You can see it here. This is the view from Scott Base. The perfect proteinase was going to have a high specific activity. That means you don't have to use much of it. We want a broad specificity so that it really chops your proteins and any contaminating enzymes that you don't want, like DNases and RNases, it breaks them down very rapidly. We wanted it to work in PCR compatible buffers because we don't want it to add anything to our extraction mixture that we're going to have to get rid of at the end of the extraction. It works without ionic detergents, and that means that it has to have that denaturation that you get from the ionic detergents, but you can do it with heat instead of using detergents. And it works at these, which is what the next line says, it works at temperatures where the proteins are denatured. As soon as you denature a protein, it becomes massively more susceptible to proteolytic cleavage. And so above 65 degrees C, proteinases, thermophilic proteinases, get a jump of about 50-fold in their specific activity. And in some cases where you have a very recalcitrant enzyme that you're trying to remove, a typical example is RNase A, mesophilic proteinases can't touch it. But as soon as you open up the structure of it by heating it up, it then becomes a target for proteinases. And the other advantage of extracting nucleic acids at a high temperature is that your nucleases are already inactive. So you're not in a situation where you're extracting DNA in a soup of nucleases and losing, fragmenting your DNA, damaging your RNA in the process. The final thing that was important to us, we didn't want to have to get rid of this with a phenol extraction, so we wanted it to be thermostable, but not so thermostable that we couldn't give a final heat at the end and get rid of it. So effectively what we did was miss out all of these steps. By removing the SDS, you could treat your sample with EA1 proteinase, heat up to 75, and because everything in that mixture is compatible, you can go straight into PCR or you can go straight into whatever your downstream application is. Now, this doesn't work for everything because, of course, in some cases, you will have inhibitory substances in your original sample, but in most cases, you don't. So this works great with most bacteria. It works well with buccal swabs, small amounts of blood, tissue samples, and there's no need for purification. Now, one of the things that you're leaving in there is short peptides. That's from the proteolytic cleavage of all those proteins in there. Now, are they going to cause problems? Well, the answer is no. We have this irony that we go through all of these purification steps to remove protein. We remove them with phenol extractions, with proteinases. We then do a 260, 280 nanometer a diagnostic on the DNA to find quantity, and we judge the quality by the lack of protein that is in there, by the 280 peak. And then the next thing we do is add BSA to our solutions to assist the downstream reactions. Most enzyme reactions in DNA, you usually put BSA or some other agent in there, which you've been diligent in removing in the preparation steps. So again, it's this dogma. Why are you removing all of the protein and the peptides if you're only going to add it back at the end? There's something else that's interesting about using thermophilic proteinases. There's actually a whole field of study in enzymology where people are trying to get one pot enzyme mixes that they can do biotransformations of biological processes with all of the enzymes in one mix. Very often you can't do that because the enzymes are incompatible. And a good example of that is when you put a proteinase in with another enzyme. The proteinase will cannibalize the other enzymes and destroy it. That doesn't mean people don't do it, but quite often you have to ask the question why. And a typical one is that many of the procedures that are out there, and these have come from many artists, are where you use proteinase K and lysozyme. Now, what happens immediately with proteinase K and lysozyme is the proteinase K destroys the lysozyme at the 37 degrees C that you're expecting lysozyme activity. This is extremely rapid. We've tested this and we couldn't even detect lysozyme in the 0 time point. So there was absolutely no purpose in putting lysozyme in that mix with proteinase K. Now we've got something a little different. We've got this proteinase that doesn't work at the same temperature that lysozyme works at. It's effectively deep frozen at 37 degrees C. In fact, the specific activity of EA1 proteinase at 37 degrees C is 0.6% of its specific activity at 75 degrees C. So it's essentially inert. So you can add your bacterial cells with a mixture of EA1 proteinase, buffer, and some mild non-ionic surfactants, and the lysozyme can play its role and it can take its part. So for the gram-positives, it can take off the outer cell walls. You then heat the mixture up to 75 degrees C, and the EA1 breaks down the cells, removes the proteinases, strips the nuclear proteins off the DNA, and destroys the lysozyme. And that's critical because lysozyme is quite inhibitory to the PCR reaction. And because we've chosen a proteinase that's thermophilic, but not so robust that we can't destroy it, you then heat up to 95 degrees C, and the EA1 proteinase is gone. So what we have then is a mixture that is relatively crude, but it's a mixture that is perfectly compatible with most substrates that you might be working on in the laboratory trying to get nucleic acids out of. So we have these, essentially with our chemistry, we have a 2-step method or a 3-step method. If we don't have an additional enzyme, we're just breaking down mammalian cells, insects, saliva, the forensic samples as well. We go straight to 75 degrees C, wait for 5 minutes, let the proteinase do its job, go up to 95 degrees C, destroy the proteinase, cool it down, and your DNA is ready. And of course, this is all closed tube. It's hands off. You're not going to contaminate your DNA with extraneous DNA coming in. You're not going to mix up your samples quite so easily. And crucially, there are so few steps that your yields are good. And this makes it perfectly suited for small samples of single cell work. For other samples, we might need a little bit extra than just the proteinase, where we add other cocktails of enzymes. It might be lysozyme, or we have some proprietary cocktails of proteinases and cell wall degrading complexes that we also use in those mixtures. And these are mesophilic enzymes that won't be destroyed by the EA1 because at their working temperatures, the EA1 is inactive. Now, we decided a few years ago to take this one stage further, and this is the machine that Joe will be talking about, and that is, yes, we have our DNA, and yes, it works well for most substances, but it didn't work well for all substances, because if you've got inhibitory substances in there, like you might have with stool sample or soil or larger volumes of blood, then you need some form of purification. So we created this device that allows us to both extract the DNA and then also purify the DNA, at least purify out the contaminating substances that are going to cause you problems downstream. So effectively what this machine does is you carry out your thermal reactions. You go up to 70 degrees C, or you do your two-step reactions at 35, 75, and then you heat it a little bit further. And what we have is some heat deformable plastic, which is a form of heat shrink tube, which bursts a heat burstable valve so that at that higher temperature, it pushes the DNA through a purification out through an exit nozzle. And what you have is your DNA in a tray at the bottom, up to 24 samples at a time. And these are purified to the extent that those inhibitors don't create any problems for you. So this we call the PDQAX, which is the pretty damn quick extraction. It was a sort of a working name that seemed to stick. And the other nice thing about this is that the purification polymer we've used also removes the EA1 proteinase. Now, if you recall, if we just use the chemistry methods, we heat up to 95 to destroy the EA1 proteinase. That means your DNA is rendered single-stranded. It's great for PCR, but if you have to make libraries, sequencing libraries, you want to do ligations, you need double-stranded DNA. With the purification polymer, that removes the proteinase instead of a heat step. We don't need a heat step, so we have nice application-ready DNA. So when we compare the methods to other steps, so if we use the PDQEX, it's hands off. You can see here where we've marked with arrows where you open the tubes. Compare it with a column or a magnetic bead method. This is easy, it's hands off. It's sometimes ridiculously easy and difficult to convince people that it will work, but it does work extraordinarily well. All of these other steps are long-winded, they take up your time, they risk contamination, and at each step you lose some yield. Now, what I'm going to do now is hand over to Joe.
Okay.
I will stop my share. Joe's going to talk about how we use the PDQEX for the DNA sequencing of bacteria.
So, right. So David's basically given the history of where the enzymes come from and the philosophy behind basically revisiting the entire extraction process and doing away with all of those wonderful additives that worked great in the 70s and the 80s, but were actually giving us so much problems. So now we're getting to the point where PDQEX will deliver beautiful DNA ready for downstream applications. But we have to show that nothing in the buffers or the presentation of that DNA, any of the proteins or peptides that may be coming through, we really want to prove that they're not going to be inhibitory to downstream processes. And one of the most important things that has come out in the recent years is the use of next generation high throughput sequencing technology. And that's really where I've got my background. So I've been using different platforms from pretty much when they became available to sequence DNA and RNA. So I was thinking, well, can the PDQEX DNA be used directly in next-gen and high-throughput sequencing technologies? So we designed a couple of experiments, and these have actually been published. So if you want to get more detail about what I speak about, there's a paper that's come out in Genes and another one in Biotechnique. So I recommend that you have a look at those as well. So we needed to say, well, what sort of sample types can we work with, and can we then do sequencing directly from the PDQEX output? And in my lab, we've tried bacteria, plants, insects, fungi, and so forth. We've also done some blood. But today I'll just talk about two studies, one based on bacteria and the other on plant and insect sequencing. And the plant and insect sequencing tells you what simplifying a DNA extraction technology can do, how liberating it can be for your science. So let's see if I can advance. So sample types we've tried. Are these advancing, David? I don't seem to be able to make the slides move.
Hey, Joe, in the lower left-hand corner, there's some arrows there.
Yeah, got it. It's just a bit slow. It's got to run around Dunedin before it changes the slides. So the first study I'll talk about is the one that we published in Biotechniques last year. Basically what we did was we grew up bacterial cultures. So we blew up some, and we split those in half. So we grew up bacterial cultures on solid agar, took 2 loopfuls of the culture and basically resuspended in 200 microliters of just plain old water, separated those into 2 even aliquots, 100 microliters each, ran the PDQ-EX extraction protocol, and we also compared it to the QIAGEN D&EASY blood and tissue extraction. And you can see immediately that when you run that extraction method, if you've got gram positives or gram negatives, it actually affects the workflow that you have to follow. So whereas in the PDQ-EX, we didn't have to worry about that so much. Okay. See if this changes. Okay, I seem to have lost a slide. Yes, okay, so these are the organisms that we used. These are clones, all extracted from different locations in Fiji and New Zealand. They had been, the species had been determined just through standard 16S. So basically knew what species group they belonged to, and we had a mix of gram positives and gram negatives. We also selected one that had a real funky pH optima because we thought that would be fun to see what it was like. So we grew up our DNA, extracted it with PDQ-EX. We did multiple cultures, and these marked with asterisks on the gel are the ones we selected for downstream sequencing. And you can see that we were getting good DNAs. They weren't all smeared. They looked good on a basic 1% agarose gel. Okay. We then did some ion torrent sequencing, which is a platform that I run in my laboratory. No changes to the protocols for making libraries. They were just standard as recommended by Life Technologies. But what we did do is the DNA extractions were done in one location, and in my lab we were just sent DNA samples that were coded. So we didn't know what organism it belonged to, and we didn't know whether it was PDQ-EX or QIAGEN extracted. So we kept, the sequencing team was in the dark, and we only broke that code at the end of the process. So basically this is a table that basically says that here are our organisms, and we've got different library barcodes that we associated with each of the DNAs, and we kept it reasonably simple. We've got a DNA quantification using qubit for PDQ-EX versus the QIAGEN. And there's a bit of difference, but by and large, these are delivering about the same quantity of DNA from each extraction, from each 100 microliter aliquot. We ran our sequencing, and this is just a table of sequencing metrics for those who are really into metrics. But basically we were getting fragment lengths about the same size. We had library barcodes here so that we minimized the number of libraries and the number of barcodes used. We got reasonable numbers of raw reads, good length sequencing for the ion torrent platform. And when we look at the filtering of those reads, basically we got between 95 and 98% recovery of good quality sequence reads from our experiment. So the quants looked good, and the metrics were looking great. The next thing we did is basically we took each of our sequencing files and we did a de novo assembly. So basically we threw all of our sequence reads together and said assemble, and try and give us some assembly statistics around our sequencing output. We lost very few of our post-filtered reads from the assembly. That was quite low, and that was regardless of the extraction technology we used. We got a few thousand contigs as expected for short read technologies, good contig lengths and so forth. So it all looked reasonably similar. The next thing we did was I went out to my colleagues who were doing next-gen sequencing and I said, well, if you were given this set of metrics from a sequencing file, which pair, and I gave them in pairs, which pair would you say would be the better metrics that you would choose to take further with your analyses? So it was kind of like choose the best out of each pair. And we found that basically of the people that were in that group, the QIAGEN extracted DNA was preferred twice, and the PDQ-EX was preferred 3 times. So it was about the same. So there was no glaring difference between DNA extraction technology and the output that we were getting from ion torrent sequencing. Oh, sorry about this, guys. It's throwing me a little bit. Here we've got what we call reference mapping. So once we had done some de novo, we'd figured out what our bacteria was and the closest relatives we could find in the databases. We basically did a reference map of our filtered sequencing reads back to the genome. And the idea behind this was to see, basically at a high level, whether we were getting biasing introduced by our DNA extraction method. And if we were seeing biases, we would expect to see very dissimilar patterns of alignment between reference genomes and our sequence data sets, between the 2 different DNA extraction technologies. And you can kind of see from this figure that the patterns are the same. We don't have any glaring changes. We don't have any holes that were in the PDQ-EX data sets compared to the QIAGEN data sets. So basically our DNA extraction method was not introducing sequence bias to our DNA sequencing output, which was cool to see. So just as we were finishing up this work, we started to do some MinION sequencing. So one of those DNA extractions from PDQ-EX, we actually ran on MinION, and then we did just a quick look-see at whether we were getting very different patterns from PDQ-EX on MinION sequencing. This is new, we were new at doing this, so this isn't as pretty as it could be, and the depth could have been a bit deeper, but basically this just is a hone in on one region, and we're seeing pretty much the same pattern between our PDQ-EX extracted DNA from a PGEM and PDQ-EX extracted DNA on MinION. So we weren't getting between those 2 different platforms kind of very large performances with the DNA that we were using. So PDQ-EX, this kind of indicated to us that PDQ-EX DNA could go straight onto MinION and could go straight into ion torrent sequencing without any problems. Okay, so basically we showed with this work that PDQ-EX, it's much simpler to get your DNA, and it works just as well as other DNA extraction platforms, chemistries on next-gen sequencing platforms, both long read and short read. So the next thing that we got involved with was in-field sequencing. And the project that we teamed up with was cassava diagnosis for viral infection. Now, just to give you a bit of an overview on what that work entailed. So in East Africa, cassava mosaic virus, it's a begomovirus, it's single-stranded DNA virus. It's transmitted from whitefly transmitted by whitefly and through infected plant cuttings. Now, cassava is the staple food for most people in East Africa. So, and there is currently an epidemic of viral infection going through cassava crops. So it's very important that we can diagnose these quickly and accurately. So cassava mosaic virus, it's DNA. We've shown that PDQ-EX can extract DNA no problem. We know how the virus is being transmitted. There are, importantly, control strategies for controlling virus. You can either grow plants from virus-free cuttings, or you can control whitefly. You can also control, grow viral-resistant cassava crops. So, okay. So the problem with diagnosing cassava quickly is that the technology to do that is not relatively, not easily available in East Africa. You can take samples from a crop, and if you send them to an international facility, unfortunately, these samples aren't generally given priority, and they can degrade en route. It's also really expensive to get the testing done. And we're talking about farmers who are subsistence farmers, and really cash flow is an issue for them. They can't afford to get testing done routinely. There's also an incredible time delay. It can take up to 6 months to get turnaround on results. When you factor in travel, time, analysis, all of these things. And 6 months basically is a lost opportunity for a subsistence farmer. Once they've planted their crop, they have a very small window when they know they need to remove that crop and replant it with a resistant cultivar. If it takes too long, they can't do that within that season. And the end result of that is starvation, basically. These people cannot grow their food, they cannot take crops to market, they cannot earn an income. They have to carry a debt from one year to the next, particularly if they never find out what virus is affecting their cassava. So it leads to that poverty spiral that we're all aware of. One way of solving this problem would be to take sequencing and do it on the farm or closer to farm. Okay, and if we're going to do sequencing on farm, it needs to be very quick and it needs to be very accurate, and in order to change lives. So basically that's what we did. If you know about the MinION technology, you know that this is a portable DNA sequencer. So this is our setup, and this is what we actually did on farm. So this is our portable sequencer. This is data analysis, basically. It takes raw signal from our sequencer and converts it on the fly to base space. This is just a power pack. We have a laptop with a curated database on it where we can do localized blast. And here we have the PDQ-EX system. Now, the only reason that this system can actually be functionalized on farm is that we have PDQ-EX. And PDQ-EX is basically a closed system. So once you've got your sample, your leaf, you grind it up, you can put it in a PDQ-EX tube, put it on that device, 20 minutes later you have your DNA extracted. So this made on-farm sequencing possible. So when we were on farm, what samples did we start sequencing? Well, we sequenced tubers, leaves, bark, roots, and whitefly. Okay, so we were able to walk onto a farm and collect these samples straight away. What do? Basically, we took leaf punches, from either a leaf or a tuber or the stem, or single whiteflies, just one flight. These things are tiny, they're about 1 millimetre long. So we took a single whitefly, we ground it up with a simple plastic Eppendorf tube and a tip, and in our homogenization buffer, which is just the reaction buffers from the PDQEX, added some enhancer and basically resuspended our homogenization mix in lyophilized EA1, transferred that mix to our PDQEX cartridge, put it into the control unit, and then ran for plant 2 different temperature cycles, one for plant, one for insects. We then took 7.5 microliters of the DNA we extracted, what came out of the end of the PDQEX, directly into a MinION rapid sequencing library, and we barcoded them, those libraries. So for each of the flow cells we ran 11 samples on each flow cell. So it wasn't just one leaf, one MinION run, it was one leaf, it was 11 samples and one MinION run in the field. By doing this, we were able to sample and sequence and get a preliminary diagnosis in the field with no mains power, no internet connection, within 3 to 4 hours. So the thing that made this possible was PDQEX coupled with the MinION and the Minute. So we sampled different leaves and stems. We had 3 in-field sequencing runs, 1 in Tanzania, Uganda, and Kenya. All of the sequencing samples that we processed generated sequence data. We were able to detect cassava mosaic virus in plants with symptoms, and also in healthy plants that showed no symptoms. We did not find virus. We did find some asymptomatic plants that were grown in very close proximity to infected plants that had very low viral titers, and we were able to detect CMV virus in single whiteflies. We have since repeated this work in the Democratic Republic of Congo and also in Sierra Leone, but I won't have that data here. So it is repeatable. Here's just some of the metrics from our sequencing run of MinION, and you can see that we actually get very long reads in our raw data sets. I mean, average length, this is likely to be the result of the rapid library making process that MinION uses, because it fragments low quantity DNA quite extensively. But we are still getting sequence reads over 260,000 bases long. So long DNA reads are present in our mixes. This is just some metrics showing lengths. And if we look here, this is a Megan analysis of the output. This is just from the Tanzanian run. You can see with our samples, we're getting reasonable numbers of raw sequence reads. A lot of them are classifiable. Most of them come from the cassava plant itself, Manahot. So most, as you'd expect, because we've just taken a leaf and ground it up, most of the DNA in there is going to be from the plant. But you can also start to see that we're picking up East African cassava mosaic virus and African cassava mosaic virus. We're also picking up whitefly DNA, and that's one of the vectors, remember, that spreads the disease. 18 leaves tested, 15 of the 18 contained virus. We've got full genome reads out of PDQEX extracted DNA, and we did some other tissues. This is the first time anybody's found a single whitefly and got virus from it, so that was quite impressive to do in the field. So in summary, PDQEX is revolutionary. It's a new DNA extraction system, and it's completely compatible with next generation sequencing workflows. It takes less time than traditional DNA extraction methods. It's comparable to material extracted from Collagen on high throughput platforms. It can be run on MinION quite successfully, and since we've done that work, we've used that many more times. Yeah, so here is just some acknowledgements. This is us sequencing in Africa, and this is the team in Dunedin. So I think that's me. Happy to answer any questions about that.
Yes, thank you, David and Joe, for your presentations. So we do have some questions here. I think the first few of these are probably for David, but Joe, please chime in too. How active is this protease?
How active is the protease? That's an interesting one. If you do a comparison of proteinase K at 52 degrees C, which really is, you're going to push out the activity to its limits at 52. Any higher, it tends to die a wee bit. It is 35 times more active, specific activity-wise. So per milligram of protein, we get 35 times the activity of proteinase K, with proteinase K at its optimum of 52 and our proteinase at 75. So they're measures that are always a little bit difficult to make, because the activity is a measure of its activity over time and its decay due to temperature. So there's different factors involved, and you get slightly different results when you do long assays and short assays. But on average, it's about 35-fold that of proteinase K.
Okay, and I think this is closely related to that. The same person asked, you didn't see it on the slide, but did one of your slides say how many units of activity or how much protein the enzyme digests to peptides in a certain amount of time?
We use these are really difficult questions with proteinases. Yeah, we use a fluorescent assay based on the traditional assay uses azacasine. It's a colorimetric assay, and they are it's very difficult to do comparative work when you're using an assay system, which, for example, azacasine or phytsicasine, it's a soluble protein that you're working on, and that's an easy target. And so on that easy target, we get that 35-fold increase. On difficult targets, it's sometimes an all or nothing. So soluble proteins versus insoluble proteins, and activity in the and stability in the presence of substrate versus not. There is some variations, but I've drifted off a little bit there, I think.
No problem. Oh, thanks. Another one for you, I think, David. Can you handle samples with very dilute target DNA? Silica bind and elute allows you to concentrate DNA, but will the purification polymer concentrate DNA?
No.
Yeah, that's a good question, and the answer is no. Where we have a lot of strength, not necessarily with the PDQEX, but with the chemistry, is with very small samples. So one of our biggest users are people doing single cell work, because you're adding all of your reagents at the beginning. Everything is compatible with the downstream tests, the PCRs and the qPCRs, so you can do your extractions in sub-microliter quantities. So in those areas where you have rare cells, but you have access to those raw cells, whether they've been FACS sorted or not, then you can do it. Now, what we are looking at is the potential for concentrating the samples up front. So as a pre-step, we concentrate the samples. As it stands, you get what you put in, but if you put in very small amounts and you extract in large amounts, it's going to be diluted. So you know, there are some provisos, and we are working on prior steps to the extraction to do that concentration step. But the question is quite right. In some cases, you know, the magnetic beads and the columns will be better if you need a concentration step.
Mm-hmm. All right, very good. Another one. Why are there different temperature protocols for different sample types? For example, you temperature cycle differently for plant and insect. Can you explain why the different temperatures?
Generally speaking, we try and match the enzymes. So when we're doing plants, we very often use a cocktail of fungal hydrolases, glycoside hydrolases, that work optimally at around room temperature. Now we push that up to 35 or sometimes 30. If we're using for bacteria, we're using lysozymes, so we use that at 37. It depends on the enzymes that we're using. That's essentially it. So not all of our enzymes work at the same temperature.
Got it. So just to be clear, you can use these reagents on your own without the PDQX, right? Or I mean, just
yes,
use
you
your
can.
cycler.
Yes, you can, and certainly with the PDQEX, you know, you're working in 50 microliter volumes. So if you're working on single cells or very tiny samples, then it's not going to be appropriate. But you know, it's different methods and different solutions to a whole lot of different problems.
Mm-hmm.
Great. And Joe, this one might be for you.
Okay.
But has soil or sediment? Have you tested soil or sediment with the PDQX? And you know, so what kind of DNA yields did you get?
Yes, I haven't done soil or sediment in my lab directly, but it is possible, and from what I've heard, it works reasonably well. So, yeah. David, have you heard anything else?
Yeah, we've done some work on the PDQX. We certainly have done soil, stool, and I noticed the question that's coming up next is stool. We've done soil and stool with the reagent methods. So we haven't had the resources just yet. It's on our list of to-dos and to provide a solution, and we're happy to work with anybody that wants to try it, and we'll guide them through it. But what we tend to do with the soil and stool is we do a pre-wash step, so we elute the biological material, the bacteria, for example, away from the solids. So you can put raw samples in there, but you'll get better results and a level of concentration if you do a pre-wash step. We have a wash buffer which allows you to dislodge the biofilms, dislodge the bacteria, and remove the solid material. And then that can go into a PDQEX, or it can go into the reagents. As I say, we haven't tested it on the PDQX. We would expect it to be better because inhibition that can come through from these samples is removed. We have a variety of different polymers that we can use in the columns, but we specifically use ones that are designed to remove polyphenolics and polysaccharides, which are the problems.
Mm-hmm. Mm-hmm.
Great. And then with that, with the stool, someone was asking about that. Should the sample be fresh or frozen?
Well, do you know, we've been having this conversation this week. It's an interesting thing because it's very difficult to get across. My background was in metagenomic analysis, largely of Antarctic soils, and a common question is which method gives you the most accurate representation of the population that's in those samples. And the question is, the answer is we don't have a clue, because it's very difficult to know what the correct one is. Every method has a bias. We have heard from some of the people that use our chemistry that they get a less bias, but that could be just for their specific soil samples that they're using. If you use an aggressive method, you lose the fragile organisms. If you use a non-aggressive method, you lose the robust organisms. And then when you come to do the PCR for the analysis, the number of cycles will affect it. Will affect the recovered population structure and so on. So, you know, what we would really like to do, and we would like to work with people that are doing it, are people that are not so much doing the PCR, which has an inherent bias, but people that might be doing metagenomic sequencing directly with the PDQEX and doing some comparisons of different methods. But the commentary is that we get we get at least a more diverse population structure than some of the other methods get.
Yeah, I think it also comes down to you can't rely on one method anyway. So whether you freeze or whether you're going to keep it at its ambient temperature, I think you really do have to try both, because you're going to see disappearance of different classes of organisms. Some will just disappear with being frozen and they're lysed, and that DNA is completely lost to any sort of extraction procedure. In some cases, so you need to try both ways. It's no one can just say, oh, just freeze it and it will be okay, because I think we all know by now that that's not really the case. Yeah.
What about extracting RNA with the system?
Okay, chemistry, we've done it. That's routinely done. With the PDQEX, watch this space. We're working on it at the moment. There is intrinsically no reason why it doesn't work. We have evidence that it works well, but we haven't validated those experiments yet. So we're currently not putting it on our website that it can be used for it. But again, if you want to give it a go, give it a go. There should be no problems.
Yeah.
Great. And then I think this is the last question for time here. What has been your overall most successful type of extraction? Crop, insects, animals, microbes?
We found that it actually works very well for everything we've tried on it. So yeah, I mean, it's a case of, again, just optimizing, but we're getting very, very good yields from bacteria. We're getting reasonably good yields from plants. So yeah, could I name a organism of choice or a sample type of choice? Not at the moment. No.
If I had to pick one, it'd be single cell work in tiny volumes. That's a particular strength, not of the PDQEX, but of the chemistry. It just enables you to work in such very small quantities. So we're developing solutions for droplet PCR, for working in emulsions, that sort of area, and hopefully we'll have something later this year that will be directly targeting that.
Mm-hmm.
Wonderful.
Cool.
Well, great. So I'm going to share this last slide. So if you have any more questions, anybody out there, and just specifically technical in nature, you can reach out to their technical staff at info@microgembio.com, start that conversation. I want to thank especially Joe and David for taking time to put this presentation together. Thank all of you all for attending. And for MicroGEM, or for DMARC Biosciences, who sponsored this webinar. They specialize in advanced genomic solutions for molecular biology and protein biology. And to learn more about MicroGEM and information about purchasing, go to dmarkbio.com and search for MicroGEM, and it'll pop right up.